Today we are continuing with the pain management series, and the lecture today is about the opioid receptors, opioid receptor structures, receptor signaling, and the agonists and antagonist drug lists. Welcome everyone. This is Dr. Mubin Sayed from DrBeen.com. Today we are continuing with the pain management series, and the lecture today is about the opioid receptors, opioid receptor structures, opioid receptor signaling, and the opioid agonists and antagonist drug lists. We will not go in every single drug, and we will not go into every single function of the opioid receptors. That will be the next one. But I wanted to make sure that we can understand the opioid receptor structure, signaling, distribution, and the main drug lists that we would then handle later on. So with this, let's look at the references first. I'm going to start sharing my screen. Here is the G protein coupled receptor article, which is one of the references. This is another reference, Physiology, Signaling, and Pharmacology of Opioid Receptors and Their Ligands in GIT, Current Concept and Future Perspectives. Then untangling the complexity of opioid receptor functions. Then GIO, I for inhibitory, O for other. So G inhibitory and G other protein coupled receptors in the aging brain. We'll talk about that too. The receptors, not the aging brain. Adenylyl cyclase type 5 is an essential mediator of morphine action. So this is a very important concept to keep in mind. We will know as we look at the lecture today that opioid agonists will activate some G protein receptors that would eventually cause adenylyl cyclase enzyme to become modulated. And so the question is, there are many classes of adenylyl cyclase enzyme. So the question is, which one? Not many people know this. So it is the adenylyl cyclase type 5 enzyme that is modulated. So keep in mind, this was a rare thing to find. And then basic opioid pharmacology and update, and the most important thing, this one, Chapter 20, Opioid Analgesia and Pain Management by Tony Yaksh and Mark Wallace. This is our best book, which is the Goodman and Gilman. I mean, Goodman and Gilman is the god of pharmacology topics. So we have Goodman and Gilman as a reference as well. So with this, let's start with my drawings. So as I said before, today's lecture is more of a pharmacology beginning for the opioid lectures. So it may bore you at times, but we have to go through this. So first of all, opiates. Opiates are compounds structurally related to products found in opium. So here you can see this is an opium flower, and this is an opium poppy. And from this poppy, we extract various alkaloids. So the opiates are compounds structurally related to products found in opium. Now, what is opium? Opium is a word derived from Greek opos or opus, which means juice. So it is the juice that is derived from the resin of Papaver somniferum or opium poppy. So this little guy over here, Papaver somniferum. Then natural plant alkaloids like morphine, codeine, thebaine are present in there, and these would be called opium. And there are then synthetics as well, which are the derivatives of these. So opiates, products structurally related to opium. Opium are the derivatives or direct natural substances in the poppy. Then is the opioid. In medicine, oid usually means looking like. So opioid over here will be the substances that look like opium. So any agent that has functional and pharmacological properties of an opiate. So any substance that may be synthetic, that may be produced or found or endogenously produced in our body or exogenously produced from outside, but anything that has functional and pharmacological behavior that is similar to opiates, which are the natural opium, then that substance will be called an opioid. Of course, then we have two kinds of opioids. We have endogenous opioids that are the ones that are produced in our body naturally. We actually all produce opioids all the time. If this lecture made you happy, you will produce your own opioids or endogenous opioids. So naturally occurring substances in animals that are opioid receptor ligands. Ligand simply means binder. So anything that can bind to opioid receptor, and if it is produced within our bodies or bodies of animals, then that will be called endogenous opioid. Then are endorphins. Endorphins are endogenous opioids in general, but also are used for beta endorphins. So you would see that there are many kinds of opioids that are produced in our body. They can generally be called endorphins. So you might hear doctors talking about, hey, when you play games or when you exercise or when you're happy, you will be releasing endorphins. So generally beta endorphins or endogenous opioids, although there are many types of them. Then is narcotic. So narcotic is an interesting term, originally Greek term from narkotikos, which means anything that would cause benumbing or that would cause person to become numb, or something that would cause stupor. That means there is a broader substances that can participate in the narcotic category. This is why in legal terms, substances with abuse or addictive potentials are called narcotics. So any drug that induces sleep is actually something that is going to cause narcosis or sleep. However, it has become associated with opioids, even in legal context. So narcotic, generally sleep-inducing substances, anything. But in medical and legal context, mostly the opioids are called narcotics. Now, the endogenous opioids, these are biological peptides. Peptides mean tiny proteins, right? So these are biological peptides in brain, and not just the brain stem, spinal cord. And you would see that even immune cells, for example, macrophages, have opioid receptors on them. And you would see GIT has opioid receptors on them as well. So there are a number of cells that have opioid receptors. But we generally, because opioids are found most commonly in the brain and brain stem and the spinal cord, that is why we say opioids are biological peptides in brain that act through opioid receptors. So here, there is a little brain over here. This is an opioid, and the opioid is binding with a G-coupled receptors or receptor, G-protein coupled receptor, GPCR. G-protein coupled receptors, you would see them. If you are from medicine already, you have known that they are the most abundant receptor types. These are transmembrane receptors. I remember that when we were doing medicine, we used to call them serpentine receptors because they have 7 units or domains that cross cell membrane and they go in and out. The first one is actually out, the last one is in, and it looks like a snake with 7 units. So it is called a serpentine protein. Here, if you see here, there are 7 units that are sitting next to each other and making a little channel. The opioid actually binds on this channel at the domain number 3 and 7. The space between the domain number 3 and 7 is a small binding pocket. That is where the opioids will bind. So it's a G protein coupled receptor. The other thing that is important to keep in mind is that internally, within the cell, this G protein is then connected with a GTPase, an enzyme that can break down GTP or work with GTP. So that is alpha, G alpha. And then there is another 2 subunits, which are called beta and gamma, which are also attached there. So that is generally a structure. We will go a little more deeper later on. Now, I'm going to give you an example of how opioids are processed in our body, how we produce endogenous opioids. So opioid precursors usually are pre-pro hormone. That means there are actually 2. There is a bigger structure that has to be processed twice to finally produce an opioid molecule. So opioid precursors are protein family. So if you see here, this is not protein, this is not a spelling mistake. These are generally called protein family. They are defined by prohormone precursors. So these precursors are prohormones. And you would see that, for example, pituitary will make a prohormone, which will then be further spliced and broken up. And finally, one part of that prohormone will end up becoming an opioid molecule. There are other molecules that are produced as well. So here in our body, we have enkephalins, which are opioids, endorphins, dynorphins. Why are they named different? Why don't we just call them all opioids? Because they all are derived from various prohormone subfamilies. So based on which subfamily these are derived from, that is how their class name is different. So we have enkephalins, we have endorphins, we have dynorphins. These are all made from different families of prohormones. I'll give you one example. This one example is POMC, which is pro, P standing for pro, OMC standing for opiomelanocortin. So you can probably see in the name, will make something opioid-like. Then it will also be precursor for melanin-type substances and adrenocorticotropic hormone. So cortin is there as well. So this is one peptide here, which will actually give rise to multiple kind of molecules. One part of those molecules here is going to become a beta endorphin. So we call this, the original hormone that is produced in pituitary is called pro-OMC. Now here, I just have a high level, a summarized illustration. This is a peptide that is produced in pituitary. If you see here in the beginning, there is a signal peptide part. Then there are various parts here. For example, this part is pro-gamma MSH. Then this is gamma MSH. This is JP, this is alpha MSLT, this is CLIP. They both together, when they are separated from the remaining structure, they would make adrenocorticotropic hormone. Then over here, we have beta LPH. And the last part of beta LPH is beta endorphin. So do we need to remember this? No. Just remember this, that we have prohormones produced in our body that would then be further sliced up. They would be further broken down into many molecules, including some opioid molecules too. So here you can see signal molecule separated first. This is the remaining part. Then this remaining part is further divided into two units. Then this last unit, beta LPH, is further divided, and this becomes beta endorphin. So this is just one example. All various kinds of opioids like enkephalins, endorphins, or dynorphins, they have pre-prohormones, which would then be, or prohormones, which would then be divided into smaller units. Okay, so that is that. Then is the opioid receptor types. So generally, we have three receptor types: mu, delta, and kappa. So mu are called MOR or MOR, mu opioid receptor. Of course, then delta will be called DOR or DOR, delta opioid receptor, and kappa will be called KOR or KOR, kappa opioid receptors. Now, if you see here, these receptors, they can all bind with opioids. However, based on the kind of opioid, these receptors behave slightly differently. For example, metenkephalin will work on mu, and if the full stimulation or activation will be plus, 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 then it has plus, plus activity on it. Delta, it has plus, plus, plus. And just very quick reminder, we did this in one of the past pain management lecture. The delta opioid receptors are found more in the spinal cord and are associated with pain gating. Mu receptors are generally the most abundant receptors and are present in the higher centers, especially the basal ganglia and the cortex. The kappa receptors are also present in the higher centers, but they are also present in the brainstem. So you can actually think about it from their location, that how would they behave. Mu receptors that are at the highest centers and in basal ganglia and hippocampus, they would of course be responsible for modulating memory, cognition, then basal ganglia and the functions and sleepiness, plus descending pain modulating pathway that we have already discussed. Descending pain modulating pathway has opioids as an important neurotransmitter in them. And so their receptors are present in the higher center. On the other hand, if you think about kappa that are present in the brainstem's role, it has vital centers in there. For example, breathing center or the cardiac centers or MSs or the vomiting centers, etc. So the kappa receptors, when they are stimulated, they would cause the suppression of these centers or modulation of these centers, causing breathing suppression or cardiac depression, etc. So it really depends where the receptor is. So for example, metenkephalin works on mu and delta, leuenkephalin, mu and delta, beta endorphin. You would see that majority of the time, or as the book wrote, Goodman-Gilman, virtually all exogenous opioids or opioid drugs, they target the mu receptors, and especially they kind of behave like beta endorphin. So if you see here, beta endorphin, mu receptors, and delta receptors. Why not kappa? We don't want kappa receptors to be activated most of the time because kappa receptors would cause brainstem depression, which would then cause respiratory depression, and that can also cause cardiac depression. We don't want to touch them. So we want to have pain modulation, right? Plus euphoria. Euphoria is produced by mu in the brain. Plus, as I said before, mu is also responsible for descending pain modulating pathways. And the delta present in the spinal cord with the pain pathway. So that means if you are going to attack delta and mu, attack or bind with them, then that would give you pain modulation in the spinal cord, pain modulation at the higher centers, plus euphoria. So wouldn't we want that one? If you add kappa to that as well, then that kappa is actually reverse. It can cause dysphoria, displeasure, plus vital center depressions. So beta endorphin, please keep that in your mind. Then dynorphin, if you see here, mu and kappa, so you can now probably think already kappa would cause dysphoria. Then dynorphin B, you can see that result over here. Then alpha, nor, neo, and endorphins. So again, more on kappa, and then endomorphin, more on mu. So the point of this little slide is to, number one, show various opioids and where they act more. And then also in this discussion, I presented that we try to target mu more than anything else. But delta are also a good target because delta are participant in the pain neuronal gating mechanisms within the spinal cord. And I have discussed that mechanism before. If you remember, it was something like this. So let's say this is the neuronal terminal for sensory nerve and the first order neuron. Then let's say this is the second order neuron in the spinal cord, dorsal horn. Then we know that there are descending pathways from the brain, and then there are internuncial neurons. And we know that internuncial neuron can release opioids that can cause second order neurons to be suppressed or hyperpolarized. Then the descending pathway from the brain that can release opioids on the first order neuron terminal, hyperpolarizing that as well. And this is a pain sensory nerve. So when the pain signals are traveling through this nerve, and if we have local internuncial neurons or higher centers, let's say you start playing games, you're happy, and all of a sudden a ball hits you. Ideally, you should feel pain. The pain signal is going through this, but there are lots of opioids being released here, which would cause local pain gating, and you would not feel the pain with as much intensity as you would feel if you were not happy and playing games and exercising or producing internal enkephalines or endorphins. Okay, so let's continue. Now, opioids in plasma versus opioids in CSF. This is also very important. The opioid substances that we find in plasma are produced, or the source for these are secretory structures. What are those structures? Anterior pituitary and, for example, adrenal glands. These produce opioids and put them in blood circulation. So when you find endorphins in the circulation, these are not coming from the brain. Although you can say that this pituitary is part of the brain, but you know that anterior pituitary is not really part of the nervous system tissue. It is present there, but it is a hormonal structure. So anterior pituitary and adrenal, they produce opioids and they put them in blood circulation. So the source is secretory glands. On the other hand, if you find opioids or enkephalins or endorphins in the cerebrospinal fluid or inside the brain tissue, that is produced. These peptides are produced within the neurexis. Neurexis is the neuronal tissue. So please remember, the enkephalins present in CSF, their source is different. Enkephalins present, endorphins present in the blood or plasma, their source is different. Then a quick, once again, review of the opioid receptors. Opioid receptors, as I said before, MOR or mu opioid receptors, delta opioid receptors, kappa opioid receptors. On the chromosomes, the mu receptors are present on the large arm of chromosome 6, location 2526. And the delta is present on chromosome 1, petite arm or small arm, location 355-33. And kappa is present in chromosome number 8, large arm, location 11.23-21. So these are the chromosomal locations for the genes that help make opioid substances. Now, the opioid receptor is a G protein coupled receptor. So if you see here, one domain, second domain, third domain, fourth, fifth, sixth, and seventh domain, although my seventh domain should be moving inwards, first is out, then seventh will be in. The amino terminal, that nitrogenous terminal, is on the outer side and the carboxy terminal is on the inner side. As I said before, between the terminal number 3, let's say this is the number 3, and 7, there is a pocket where the opioid ligands or opioid substances, they would bind here. As you can see, this little opioid substance is doing. On the intracytoplasmic side or the cytoplasmic side, there are 3 proteins that are attached here. So a GTPase protein, G alpha, and then we have beta and gamma subunit. This beta and gamma subunit, they work together, and this G alpha subunit, that works alone. Now, the distribution of the opioid receptors. Number one, these are widely distributed within the nervous system. We know that. However, they are also present, these receptors, also present in non-neuronal tissues, for example, macrophages, glial cells, astrocytes, and GIT, enteric nervous system. Highest concentrations. So we're not talking about the only distributed parts, but highest concentration. Where are they found the most? The mu receptors, highest concentration, cerebellum, caudate nucleus, nucleus accumbens. So the caudate nucleus and the nucleus accumbens are part of basal ganglia. Then delta opioid receptors, hippocampus, cerebral cortex, putamen, caudate, accumbens, temporal lobe. These are all in the higher center. But as I said before as well, delta are very common within the spinal cord, especially for the pain gating. The structure names here are those for the highest concentrations. Then kappa, caudate, accumbens, putamen, and hypothalamic nuclei. Again, highest concentrations. As I said, they're present in other parts as well. So before I read you this part, if I said to you that what one thing should come to your mind when you think about mu receptors, that should be descending pain modulating pathway, number one. If I said another thing, that will be euphoria. So these are the 2 important ones to remember with mu. If I said, remember one thing, what is the most important thing for delta? So then, of course, the pain gating, 1, and then anxiolytics. Delta are very good with anxiolytic behavior as well. And then one thing about kappa. So kappa, they all have analgesic effects. So kappa are also analgesic, but kappa do cause dysphoria as well. So please remember this, kappa can cause dysphoria. Okay, so here, I wanted to read one of the paragraphs, which I think is very important. In the CNS, opioid receptors are expressed in pain-modulating descending pathway involving locus coeruleus, medulla, and periaqueductal gray matter, and are mainly involved in pain signaling and anti-nociception. Pain signaling means modulating the pain, usually towards suppressing of the pain, and anti-nociception is also the suppression of unpleasant feelings. Pain is called nociception because it is always unpleasant. But there can be more things than pain that can be unpleasant as well. So anti-nociception is one of their effects. Opioid receptors also occur in midbrain, now we're talking brainstem, limbic and cortical structures. Limbic system, our emotional system, that is how euphoria would occur. And the cortical structures, the frontal lobe, and thus not just the frontal lobe, cortex, and thus modulate a wide range of other functions, including memory and stress response. Especially the hippocampal involvement will mean memory plus cognition both. This is why there are some researches nowadays for brain fog that may be related to opioid receptor abnormalities. So memory and stress responses. Activation of opioid receptor attenuates neuronal activity by pre- and post-synaptic mechanism, which include the release of inhibitory neurotransmitter and change in neuronal excitability. So we look at the receptor behavior, but I would continue to make this little structure here. First order neuron, this is the terminal of it. Second order neuron, presynaptic terminal. This is presynaptic, this is postsynaptic. Then here is the higher center. Here is the internuncial neuron. This structure has lots of opioid receptors and releasers. So for example, higher modulation the signal from the higher centers of pain modulation will release opioid here on the presynaptic membrane. Internuncial neurons can release opioids on the postsynaptic membrane. So I'm going to say this so many times that this will become a picture that you would even dream of. So back here, activation of opioid receptors attenuates neuronal activity by pre- and postsynaptic mechanisms. So I want you to have something in your mind. How will the neuronal activity be attenuated or suppressed? I'm going to explain that a little later. I'll give you a hint right now. The inward rectifying potassium channels open up and they cause potassium to exit, hyperpolarizing the cell membranes, and calcium channel entry or calcium channels are blocked, reducing the entry of calcium, which will then reduce the amount of neurotransmitter release. Both of these have an effect of suppressing a neuronal structure. So why have I made this neuron with soma and terminal? Most of the opioid receptors are distributed on soma or the cell body of the neuron and on the terminals of the neurons. Now, last few topics. So agonist ligands. So these are the binders to the opioid receptors that are agonists that are going to behave the same way as opioids do. So this is a direct sentence from Gilman. The reason I took direct sentence from there is that I am not said to, sometimes people say, well, Mobeen said this happens like this and Mobeen may be wrong. So this is Goodman-Gilman's sentence. Virtually all clinically important ligands target mu opioid receptors, or clinically important ligands target mu opioid receptors. So opioid ligands, these are agonists. So etorphin, look at this, mu triple stimulation or maximum, delta maximum, kappa maximum. Fentanyl, which nowadays has become a problem, mu. Then hydromorphone, mu mainly, and then some kappa, levorphenol, mu methadone, mu morphine, mu some kappa, then sufentanil, mu some delta, some kappa, DAMGO, mu. So if you can see, this main class or list of drugs are on mu receptors. Then if you see here, bromazocane, mostly on kappa, buprenorphine, partial agonist, not complete agonist, and also suppresses kappa. Butorphanol, partial agonist and activates kappa or is agonist to it. Activation could mean something happens, although when the opioid receptors are activated, internal system or enzymes are modulated in a way that some suppression occurs. So activation should not be taken here as the cell will be activated. Activation of the receptor, so just simply calling agonist is better. So butorphanol, partial agonist plus kappa agonist, nalbuphine, antagonist to mu and agonist to kappa. DPDPE is mu agonist and USO U50488 is delta agonist. So this is one list of the drugs that are agonist. Now just a quick list of the drugs that are antagonist, and here a couple of things to keep in mind. Generalized antagonist, naltrexone and naloxone. And you would see nowadays naltrexone is becoming so important, and it has been helping not just for the addiction, but for other neuroinflammatory states as well, that it is a magical drug. So naloxone or naltrexone are pan-antagonists. They are antagonists of opioid receptors everywhere and for all receptors. Other than those two, there are actually some other antagonists as well, meaning they would block the receptors. However, most of these are for research purposes and not for administration. So naloxone and naltrexone, just please never forget these two drugs. So if you see here, naloxone, mu suppression, delta suppression, kappa suppression. Naltrexone, mu suppression, delta suppression, and more kappa suppression than naloxone. Naltrexone is very common nowadays, and low-dose naltrexone works wonders. Now, quickly, the opioid receptor signaling. This is the last part of the topic here. So I would give one more quick work with how the potassium and calcium channels will affect the cell. But let's just very quickly look at how opioid receptors, when they are activated, what will they do? So here is a receptor. We already know that there is a GTPase subunit and beta and gamma subunits. So here is what happens. Imagine This is the delta receptor, opioid receptor, and this is mu opioid receptor. Here is a kappa opioid receptor. So let's first look at the delta and mu. When these are activated, when these bind with their ligand, agonist ligand, if you see here, I've written GIO. So these are G proteins which are inhibitory or other. So G proteins are classified as stimulatory, inhibitory, etc., right? So there are certain G proteins for which we're really not clear for their action. And so we just call it an other, other action. So GIO simply means inhibitory and other. So here is what happens. When these mu or delta G proteins are activated by agonist, then what happens is that the G part, so this part, this separates, and the beta gamma part, that separates as well. So number one, they separate from here. Number two, they separate from each other as well. Beta gamma stay as one unit and G becomes separate unit, or this is alpha. So G alpha becomes separate. Now what happens is, if you see here, when there is an agonist, then the alpha unit becomes active and it produces GTP. That causes a reduction in adenylyl cyclase, which in turn causes reduced cyclic AMP. I shouldn't say reduction, it just modulates it, but reduction in adenylyl cyclase, which then reduces cyclic AMP and pKa activation. And that has many, many effects. That is why if you see here, I've not written what is the end effect, because this branch would have many internal functions. And as I said before, one important function to keep in mind, the chronicity of the opioid behavior is driven by this branch. Now, as these two were any one of them was activated, this we just saw that what would alpha unit do? The beta and gamma unit, here they are. When they become activated, they cause inward rectifying potassium channel to open up, causing potassium to get out of the cell, hyperpolarizing the cell and stabilizing it. And then secondly, the calcium channels are blocked, so calcium cannot get into the nerve ending and the nerve cannot release neurotransmitters. And so that nerve is kind of suppressed and the receiving nerve or the postsynaptic membrane is stabilized. Why? Because it is not getting sufficient neurotransmitters. So it's a suppressive behavior, both of these. Then if you look at kappa, wherever these are present, when kappa is activated, again, it has the same structure. It has an alpha subunit and beta and gamma subunit. Alpha subunit would do the same thing as this alpha does, and that is reduce the adenylyl cyclase. The beta gamma subunit would cause calcium channel blockade, which would then cause reduced release of neurotransmitter. So that is the discussion. But if you have one more minute, I want to very quickly explain how the potassium channel and calcium channel's behavior acts on the nerve. So let's say we have a pain nerve coming from some part, and this is the ending of it. So if I made a neuron here, let's say this is a neuron, although pain neurons are pseudo-unipolar, this one is multipolar. So let me make it a correct pain neuron. Let's say we have this pain neuron, cell body. This is the one of the axon central part. This is the axon peripheral part. Let's say these are free nerve endings. There is some tissue over here from which the pain is going. This is the neuron cell body. This is the nerve terminal. This nerve terminal will end up in the spinal cord, right? So if there was a spinal cord over here, it would end up in the dorsal horn of the spinal cord. Over here, it would relay on a second order neuron, correct? Now, what happens is when the signal, so let's say there is something happened here, which would cause a pain signal to start and go all the way here. It would reach here, then the neurotransmitters will be released, and the second order will become activated, and the pain signal would go all the way to the brain. Good. Now, if we can suppress the action potentials in this nerve, then we would have stabilized this nerve or gated the pain signal. And if we can, even when the pain signal is able to arrive here, if we suppress the production of neurotransmitter, not production, release of neurotransmitter, that is going to work on the postsynaptic membrane, then we have also stabilized this activity, and the pain signal would come all the way like a wave coming to the shore and attack here and then die. So how does that happen? First, potassium channels. So inward rectifying potassium channels, when they are activated by opioids, as we saw here, this one. So when these are activated, they cause potassium to get out. When the potassium is moved out of the cell, then the cell inside becomes more negative. We call that as a hyperpolarized state. When the cell is hyperpolarized, it is difficult to create action potentials in the cell. So even when the pain signal is arriving here, it is going to come here and kind of die out. Or we would need more intensity and more frequency of the pain signals to create a signal that can continue on. So that is one behavior, hyperpolarization. Now, this can happen on the post-neuronal side as well. And that would also cause hyperpolarization here. So now this guy, the first order neuron, is already sending or filtering or is suppressed already for various signals. And if some signals are passing through it, they are now dying here in the second order neurons beginning, because that also is hyperpolarized. So both cells are stabilized and less activity or pain activity is happening, that is analgesic effect. And if it is in the brain, then euphoria, depending upon where the receptor is. I'm using spinal cord. Now, the second part is the calcium. So here on the neuronal terminal, we have calcium channels as well. Calcium channels are really important in the neuronal tissue because when we want to release a neurotransmitter, which would be released from one neuron and will act on the other neuron, this release needs the calcium to come into the cell, and then calcium would help to drag, or calcium would activate the proteins that would drag the neurotransmitter vesicle and bring it to the cell surface so that the neurotransmitters can be released, right? So if the calcium entry is less, then the neurotransmitter is actually sitting there. For example, in the pain nerves, it is substance P. So the substance P is sitting there. It can actually go to the next one and cause the activation and signal to continue. But if there is less calcium coming in, then there is not enough calcium to activate the proteins that would drag this vesicle to the surface and have the neurotransmitter be released in the synapse and go to the next one and cause the signal to continue. So all of a sudden, neurotransmitter is reduced even when the pain signals are coming here to this area. So what a wonderful mechanism. One, we have stabilized both cells. They're already kind of responsive less. And then on top of it, we are blocking the neurotransmitter release. And the end result of that will be pain modulation. If it is spinal cord, if it is cortex, then euphoria and the descending pain modulation. If it is brainstem, then of course we know that the effect on the brainstem cells. Okay, so this is the discussion